At the end of this practical session, the students should be able to explain the principle of ELISA, to demonstrate the technique of ELISA, to discuss about its applications, and to interpret the clinical conditions correlated with their laboratory investigations by ELISA. Okay, so before starting about the principal instrumentation or the demonstration about the ELISA, can you please tell me, have you heard this name before this? Do you have any idea? कहीं आपने ये टर्म सुनी है ELISA की कि ELISA क्या होता है, कब इस्तेमाल होता है, कहाँ इस्तेमाल होता है? नहीं, ठीक है, वेरी गुड. अच्छा जी, कहीं आप लोगों ने have you ever listened about to perform a blood test for the presence or for the detection of some antibodies? कभी ऐसा सुना है कि किसी ने आपसे कहा हो कि अपना एंटीबॉडीज टेस्ट करवा लो किसी भी डिसऑर्डर के लिए? सुना है. सबसे ज्यादा कॉमन सिचुएशन जो करंट हमारे पास जो पेंडेमिक सिचुएशन है इस कोविड की, मोस्टली हमारे पास एक तो कोविड का डिटेक्शन के लिए हमारे पास टेस्ट होता है, लेकिन समटाइम्स हमारे पास कोविड रिकवरी के बाद कुछ ऐसे भी पेशेंट्स होते हैं जिनके बारे में कहा जाता है कि जी तुम्हारी एंटीबॉडीज कोविड की बन चुकी हैं, नहीं बन चुकी हैं, एंटीबॉडीज हैं, एंटीबॉडीज नहीं है. ऐसा कुछ कभी सुनने में आया है? कोविड एंटीबॉडीज का टेस्ट करवा लो, पता लग जाएगा एंटीबॉडीज अभी बॉडी में मौजूद हैं कि नहीं है. अगर एंटीबॉडीज हैं तो यू आर सेफ, अगर एंटीबॉडीज नहीं है तो यू आर अनसेफ. ऐसा सुना है? तो जनाब ये जो एलाइजा का हमने वर्ड सुना है जो हमें बहुत ही मुश्किल सा लग रहा है कि जी पता नहीं ये क्या है एलाइजा. ये एलाइजा वही है जिसके बारे में हम तकरीबन रोज ही सुन रहे होते हैं इन द फॉर्म ऑफ कोविड एंटीबॉडीज. तो जो कोविड की एंटीबॉडीज हमारे पास डिटेक्ट हो रही होती हैं वो कैसी होती है? थ्रू एलाइजा. दिस इज द इंस्ट्रूमेंट ऑफ एलाइजा. Somehow it is similar with spectrophotometer, but it is not an spectrophotometer. This is very different from spectrophotometer. The principle is different. So ELISA, the combination of enzyme linked immunosorbent assay, ELISA, ELISA. This is an immunological assay. The term ELISA was first used by Engvall and Perma in the year 1971 while they are working on the detection of HIV, human immunodeficiency virus. So the first identified virus by ELISA was HIV. So this ELISA is not only used for the detection of antibodies, it can also used for the detection of antigen, because this HIV is antigen, it is not antibody. The antibodies are present against the antigens in our body. So the ELISA techniques, it is commonly used for estimation of different antibodies, antigens, cytokines, proteins like glycoprotein, hormones, receptor proteins, to screen forensic specimens for drugs of abuse and toxicology of the drug. The side effects or the toxic effects or the harmful effects of the drugs can also be identified by using the ELISA. It is highly sensitive process, can detect proteins even at the picomolar or nanomolar concentration. The sample that is used, usually it is a blood sample or it may be serum or plasma that is made from the blood, or sometimes a urine sample can also be used for the diagnosis. Why this instrument is called ELISA? As its name indicates, enzyme linked immunosorbent assay. Antigen or antibody of interest is absorbed onto the plastic surface of ELISA. This is called as sorbent. Antigen is recognized by a specific antibody, so it is immunology. This antibody, which is recognized by the antigen antibody, the antibody is linked with an enzyme, so it is enzyme linked. Substrate reacts with enzyme to produce product. Usually this substrate is colored and read by spectrophotometer. So this is an enzyme-linked immunosorbent assay in which the antigen and antibody complex, which is formed by the antigen-antibody reaction, it is linked with an enzyme. So this is enzyme-linked immunosorbent assay. Sorbent means the antigen are absorbed on the plastic surface of the ELISA. Then we add the specific substrate of that enzyme. The substrate starts to convert into the product. The concentration of the product shows the concentration of antigen-antibody complex. So again, this color complex, which is produced at the end of the reaction, it is read under the spectrophotometer, the colorimetry. The intensity of the color depends on the concentration of the sample. ELISA is a sensitive immunoassay test that uses an enzyme linked to an antibody or sometimes an antigen as a marker for detection of specific proteins. Quantitative analysis is usually based on detection of intensity of transmitted light by spectrophotometry at specific wavelength. The two main variations on ELISA are: it can be used to detect presence of antigens that are recognized by antibodies. If we are going to perform a test for detection of antigens, then our sample contains antigen which are coated or which are absorbed on the plastic surface within the ELISA. Then we add enzyme-linked antibody that binds with the antigen present in the blood. Sometimes we want to detect the presence of the antibodies. Then if we are going to perform estimation of antibodies, then what we will do? We apply our sample that contains antibodies on the plastic surface. Then we add antigen that are coated or that are linked with the enzymes. So we can detect antigen as well as we can detect antibodies. If we are going to detect antigen, then we add enzyme-linked antibody. And if we are going to detect antibodies, then we are going to add enzyme-linked antigen. So at the end, the antigen-antibody complex is formed, and the enzyme that is linked with this complex, it starts to convert its substrate into the product. And the color is produced at the end of the reaction that is read under the spectrophotometer, which is present inside the ELISA or which is attached with the ELISA. The applications of this instrument for the detection of HIV, detection of dengue virus, hepatitis virus, rotavirus, malaria, and some other infectious disorders, tuberculosis, COVID antibodies after the recovery from COVID or after the vaccination. So if you want to check whether the vaccine that you have faced, it is whether it is working or not, you can perform your antibodies test. The antibody detection, whether for COVID, whether for HIV, whether for any other infectious disorder, all of the antibodies are detected through ELISA. Human T-cell leukemia virus type 1, it is also detected by ELISA. Antiparietal cell antibodies that are produced during the condition of gastritis because of the infection of H. pylori, Helicobacter pylori, a bacteria that can cause gastritis, and pernicious anemia. In both conditions, antiparietal cell antibodies are formed that can be detected by ELISA. Squamous cell carcinoma, a type of skin damage or skin cancer because of extra exposure to UV light or radiations. Syphilis and chlamydia. These are the infections associated with sexual contact. Lyme disease and Rocky Mountain spotted fever. These are the bacterial infections usually caused by the insect bites. Okay, now there are different types of the ELISA. direct ELISA, indirect ELISA, sandwich ELISA, competitive ELISA. I will explain you in detail at the end of the session. The basic principle of all types of the ELISA is same. The basic theme is same. There is antigen antibody reaction, which is linked with enzyme. Then we add a substrate, which is specific for the enzyme. This enzyme starts to convert the substrate into the product. At the end, the result is obtained by reading at spectrophotometer at the specific wavelength of that reaction. This is the basic theme of all these 4 types of the ELISA. There is a little bit difference. What is the difference? I will tell you it at the end. Okay, there are some specific terms used while performing on ELISA or while reading upon about the ELISA. Solid phase, 8 centimeter into 10 centimeter plastic plate which contains 8 into 12 matrix of 96 wells. If you can see here, this is a plastic plate that contains 96 portions called as wells. 96 wells. Each well is about 1 centimeter in height and 0.7 centimeter in diameter. So this plate contains 96 columns or 96 wells. So it means we can perform ELISA test on different samples at the same time. Out of these 96 wells, as it reads under spectrophotometer, so we use a blank, we use 4 to 5 standards, and the rest of the wells are for our sample. So we can use or we can run more than 50 or around 90 samples at a time, because this plate contains 96 wells. Out of these 96, blank and standards are prepared or they are added into the wells, and the rest of the wells are for the test samples. This test sample could be blood, serum, plasma, or sometimes urine. Adsorption, the process of adding an antigen or antibody diluted in buffer, so it attached to the solid phase on incubation. The sample that is to be analyzed, it is adsorbed on this microplate. So this is the process of adsorption. Then washing, the simple flooding and emptying of wells with a buffered solution, the washing of extra antigens or extra sample from the wells. Once we add the sample on into the well of this microplate, then this sample is allowed to adsorb onto the surface of this well, on the surface of this plate. Then after a particular time, the rest of the sample is washed through a washing buffer. Antigen is a molecule or a foreign particle that elicits the production of antibodies when introduced into the body. Antibodies are the proteins that are produced in response to an antigen. Enzyme conjugate, an enzyme that is attached irreversibly to an antibody. Mostly the enzyme which is used in ELISA is horseradish peroxidase enzyme. This is an enzyme which is present in the roots of a plant horseradish. It is highly stable enzyme which is present only in plants, not in animals. So this enzyme is linked with the antibody that is to be added into the microplate that binds with the antigen. If the antigen is present, then antigen antibody reaction is going to take place. Chromogen is a chemical that alters color as a result of an enzyme substrate reaction. Mostly we use a color reagent or a chromogen trimethylbenzidine for ELISA, TMB. Stopping solution or stopping of the reaction. To stop the reaction, Then reading, color complex is read by spectrophotometer at a specific wavelength. So these are the some terms that are used in ELISA. The principle and the requirements. Principle is based on antigen-antibody reaction, which is linked with an enzyme. Sample and standards that contain antigen for a specific condition is added on the microplate in different wells. So here we should have to mark the well. Okay. The marking is very important. In which well we have which sample. Respective antibody which is linked with an enzyme is added on the plate. A specific substrate of that enzyme is added that binds only with enzyme which is present as enzyme antibody antigen complex. Enzyme substrate complex is formed and product is produced. The reaction which is catalyzed by the enzyme is stopped by adding the concentrated acid, which is called as stop reagent. The color complex which is produced is treated with the reagent or a chromogen, and this complex is read by spectrophotometer at a specific wavelength. Requirements: polystyrene microplate of 96 wells. We require our sample, test sample, standard sample of known concentration to relate with the sample to calculate the concentration of the molecule of interest into the sample. Washing and coating buffers. Antibodies linked with enzyme. Substrate of that enzyme. Stop reagent which is usually concentrated H2SO4. Color reagent which is TMB. The reagents that are used: coating buffer, which is usually phosphate buffer of pH 7.4. Washing buffer, again it is a phosphate buffer. Blocking buffer, usually it is bovine serum albumin. The enzyme system, usually it is horseradish peroxidase. As I told you earlier, it is an enzyme which is present in plants, the root of a plant horseradish. Or second option is alkaline phosphatase. So the substrate, if the enzyme is horseradish peroxidase, then the substrate is OPD, O-phenyl phenylene diamine dihydrochloride, OPD. Phenylene diamine dihydrochloride, that gives yellow-orange color, and this color complex is read at 490 nanometer. If the enzyme is used alkaline phosphatase, then the substrate is PNPP, para-nitrophenyl phosphate. Again, it gives yellow color, but the intensity of color is different. So it is read at 405 nanometer. So for enzyme HRP, the substrate is OPD. For enzyme ALP, the substrate is PNPP. The color reagent usually used is trimethylbenzidine, TMB. And to stop the reaction, we use concentrated acid, H2SO4. Procedure: a solid plate or a microplate on which the antigen is coated. After coating of the antigen, this antigen is washed by coating buffer and washing buffer. After a period of incubation, then we add an antibody specific for that antigen. This antibody is linked with the enzyme. Then again we incubate it. We give the time to make antigen-antibody complex. Then after a particular time, we add a substrate of that enzyme. At the end of the reaction, product is produced. Chromogen or a color reagent is added to give a yellow color. and H2SO4 is added to stop the reaction. Then the color produced or the color compound is detected or it is read by a spectrophotometer at 490 nanometer if we used HRP enzyme, or at 405 nanometer if we used ALP enzyme. But usually we use HRP, horseradish peroxidase enzyme. So the first step is the coating of plate with coating buffer and antigen. Then we remove extra liquid by washing. Blocking and unrelated protein-based solution is used to cover all unbound sites on the plate to remove the other impurities or to remove the other proteins from our sample. The blocking or the washing or the coating of the plates will be done. And after the washing, blocking, and removing all other additional non-protein or other interfering molecules. Then we add antibody, which is linked with the enzyme. The enzyme conjugate antibody or antigen binds specifically to the target antigen or antibody. Again, by the step of washing, after adding the antibody, which is linked with the enzyme, there is the second step for washing. The first washing was done after the addition of the sample, and the second washing was done after the addition of the enzyme linked antibody to remove extra or unbound enzyme from the sample. Then the results will be read under the spectrophotometer. For example, this is the yellow color developed. If the result is positive, then it gives a yellow color, and if the result is negative, then the color is not produced. So if our sample contains antigen, so the antibodies binds with the antigen. And if the sample does not contain any antigen, then the antibodies that are linked with the enzyme, which is added into the wells. After washing, all of the antibodies along with the enzyme washed away from the wells. If we add the substrate, if we add the color reagent, if we add the stop solution, we don't obtain any result because the antigen antibody complex is not formed. So the yellow color indicates the presence of the antigen or sometimes presence of the antibodies. This is the ELISA microplate reader. The microplates is inserted into the ELISA that contains the spectrophotometer. Again, the principle is same that you have already done in spectrophotometry, and the result is obtained in the form of the hard copy sometimes, depending upon the type of the instrument. Sometimes it gives a digital reading onto the screen, and we would have to plot the standard curve against the concentration and absorbance, just like a spectrophotometer. So in qualitative estimations, we get positive or negative results. The positive results indicates the presence of the antigen or presence of the antibody. For quantitative results, the optical density or the absorption or the fluorescent units of the sample is incorporated into a standard curve that gives the severity of the infection, the concentration of the antibodies present in the sample, or the amplitude of the infection. So the qualitative analysis through ELISA indicates the presence of antigen or antibody, whether this is present or not, and the quantitative estimation gives the quantity, the amount of the antibodies present in the sample. Then we plot the standard curve of concentrations on the x-axis versus the absorbance of the standards on the y-axis. Concentration of the blood sample was calculated from the standard curve. For example, this is a standard curve. Absorbance versus concentration of standards. Then by this standard curve, we can calculate the concentration of our sample. Sometimes we obtain this standard curve from the ELISA in the hard copy form. And sometimes we should have to plot this is standard curve by ourselves, depending upon the type of the instrument. The modified is the most modified instrument gives the most modified result. Okay, now before starting the precautions, I would like to share a video with you. Please listen and watch it carefully.
Hello friends, in this animation I am going to explore enzyme-linked immunosorbent assay. ELISA is used to detect antigens of many diseases like AIDS, tuberculosis, malaria, etc. ELISA is based on the principle of reaction between enzyme and the substrate to detect the antigen present in the sample. So, antigen will be taken and the enzyme-linked antibody will be added, which will form an antigen antibody complex. Now we will add specified substrate for that enzyme and gives result in the form of color change due to enzyme substrate reaction. Now coming to the methodology of ELISA. First we will take a polystyrene 96 wells microtiter plate, then add the sample which contains antigen. The sample may be patient's blood or serum or any other part. So antigen will attach to the surface of the wells of microtiter plate. Now wash the microtiter plate by using
washing, coating and washing
of pH 7.4 and 0.01 to 0.05% of twin 20 detergent solution. It will remove unwanted materials from the wells of microtiter plate. After that, add specified enzyme-linked antibody to the wells of the microtiter plate, which will form antigen antibody complex. Now again was the microtiter
second
plate
step
by
washing
using same buffer solution like this.
to remove extra
Washing specified
enzyme.
substrate for the enzyme is added and the enzyme substrate reaction takes place, which forms blue color in general. Now add acid like H2SO4. So color of this solution in microtiter plate becomes yellow. So at the end, this yellow colored solution will be observed under colorimeter in a specified wavelength and the absorbance is determined, and we can conclude that the ELISA test is positive or negative. Now coming to the different types of ELISA. So
now listen and see the types of the ELISA
carried out, which are direct ELISA, indirect ELISA, sandwich ELISA, and the competitive ELISA. Now coming to the direct ELISA, which is already discussed in the methodology. So first we will take a sample which contains antigen, then we will add enzyme-linked antibody which forms complex with antigen like this. Now add the specified substrate for the enzyme used and both will react and forms a product which is blue in color like this. Now coming to the second type of ELISA, which is indirect ELISA. So in this type of ELISA, we will take the sample and then add antibody which will form an antigen antibody complex. Now we will add an enzyme linked secondary antibody which will react with the primary antibody and finally specified substrate is added. So substrate will react with the enzyme and again forms blue color like this.
So it means in indirect type of ELISA, we use two types of the antibodies, the primary antibody and the secondary antibody. Primary antibody binds with the antigen present in the sample and the secondary antibody is linked with the enzyme. This is indirect ELISA.
Now coming to the sandwich ELISA. In this ELISA, we will take a polystyrene microtiter plate and the surface of that plate is coated with antibody.
The surface is already coated with antibody of interest. Before adding the sample, this microplate already contains antibodies.
Add the sample containing antigen.
This is the sample.
Again add enzyme linked antibody.
So this is a sandwich.
So, antigen will be sandwiched between two antibody layers. Now we will add a specified substrate which will react with the enzyme and gives blue color. Now coming to the competitive ELISA, which is used for very low concentration of antigens. In this type of ELISA, we will take antigen
As the name indicates, there is a competition. So even if a protein is present at very lower concentration, we used some antagonist or some opposite molecules to compete this molecule for the binding. So this is the competitive ELISA.
Antibody in a separate vessel. Now this solution contains antigen antibody complex and the free antibodies. After that, we will take a microtiter plate which already contains a specified amount of antigen B. Now we will add the solution to the microtiter plate, so remaining antibody will react with the antigen B which is already present in the microtiter plate. After that, washing is done, so antigen A and antibody complex will be washed out. Now enzyme linked secondary antibody will be added which will react with the primary antibody. After that, specified substrate will be added which gives blue color. Now in next step, we will add H2SO4 which leads to the appearance of yellow color. If absorbance in the colorimeter is higher, then concentration of antigen A will be lower. If absorbance in the colorimeter is lower, then concentration of antigen A is higher. For more details,
okay, so I hope now you have understood very well about the ELISA, about the principle of the ELISA and the procedure of the ELISA. Okay, now the precautions. Use separate pipette or change the tips of the pipette every time for separate solution. Wash the plates very carefully. The washing step that were done twice after adding the antigen onto the plates and then after adding of the enzyme-linked antibody. Both steps of the washing should be very carefully. Use plate cover during incubation to avoid the exposure to sunlight. Improper coating of wells may give false positive result. Okay, now there is a comparison between COVID antigen analysis and COVID antibody test. The presence of COVID antigen is usually done by PCR, polymerase chain reaction, and the presence of COVID antibodies is done by ELISA. PCR test detects whether the virus is present or not, and the antibodies test detects whether the antibodies are present or not. The antibodies cannot detect through PCR because this PCR is performed only for DNA and RNA analysis, not for proteins and carbohydrates. The sample that is used, usually it is a nose swab or throat swab, and for antibody estimation, the sample used is blood. I am talking about the COVID only, not for other disorders. Positive PCR test indicates current infection, but the positive antibody test indicates past or the previous infection, because the antibodies are synthesized in the body once the antigen attacks in our body. In response to that antigen, our body produces antibodies. Okay, so we can detect the presence of a virus or the presence of an antigen in our blood through ELISA. But what is the difference between PCR and ELISA? We can detect the presence of antigen of HIV, COVID, even COVID, like hepatitis, tuberculosis. We can detect whether this antigen is present or not through ELISA as well. But the difference between ELISA and PCR is the PCR is highly specific for DNA and RNA multiplication and reproduction. So ELISA just gives the whether this antigen is present or not, but the PCR gives the rate of multiplication of that antigen. So the PCR is different from the ELISA. The principle and the procedure is totally different. The objective of PCR and ELISA test are different, but here we are talking about the ELISA, which is used for detection of antibodies. And sometimes we can use ELISA to detect antigen also. But this ELISA does not give the multiplication rate of antigen. Advantages of ELISA: it is quick and accurate. It gives quick and accurate results. Highly sensitive. It can detect protein even at pico or nanomolar concentration. Simple to perform, easily automated, more accurate results. Compare favorably with other methods such as radioimmunoassay and another type of immunological assay, RIA, radioimmunoassay. It uses radioisotopes for immunological assay. So this ELISA is preferred over RIA because it does not use any radiological material. So it is not harmful as compared to RIA. Multiple samples can run at same time, but the condition is the samples that are going to be added into the microplate should also, they all have contains all molecules of the same interest. अगर हमें कोविड एंटीजन ही आइडेंटिफाई करना है तो हमें तमाम वेल्स में कोविड एंटीजन का ही सैंपल रखना होगा. हम ये नहीं करेंगे कि हमें हेपेटाइटिस बी का पेशेंट है, कोई हमारे पास कोविड का पेशेंट है, कोई हमारे पास ट्यूबरकुलोसिस का पेशेंट है, डेंगू वायरस मल्टीप्लिकेशन हमारे पास डेंगू वायरस की एंटीबॉडीज चेक करनी है, हम सबको एक साथ कर देंगे. नो, तमाम हमारे पास जितनी भी एंटीजेंस हैं, जितनी भी एंटीबॉडीज हैं, इन तमाम एंटीजेंस और एंटीबॉडीज का एक अपना स्पेसिफिक मैकेनिज्म है. तो हमें उन सब के लिए जो है वो एक सेपरेट हमारी किट्स आती है. ELISA for estimation of COVID antibodies, ELISA for detection of malaria antibodies, ELISA for detection of HIV, for tuberculosis, for dengue, hepatitis. तो हमें जो भी एंटीजन पे हमें काम करना है, कंडीशन ये है कि all of the sample should be of the same of interest. ऐसा हमें नहीं करना कि हमें हाफ सैंपल हमने 96 वेल्स में से हमने जो है वो 4 सैंपल कोविड के लिए लिए, 2 सैंपल HIV के लिए लिए, 10 सैंपल हमने डेंगू के लिए लिए. नो, ओके. तमाम सैंपल्स हमारे पास जो है वो सेम होने चाहिए ताकि हम उसमें जो एंटीजन एंटीबॉडीज ऐड कर रहे हैं वो हमारे सबके जो है वो सेम रिजल्ट्स दें. There are also some disadvantages of ELISA. It is highly expensive and sensitive to prepare antibodies of a specific antigen. Very sensitive to handle because of multiple washings. Weak antigen antibody is not detected. So there is a chance of wasting some antigen and antibodies if their complex is very weak because of multiple washing. It takes 4 to 6 hours for detection, including washing, incubation, reading, detection, calculation. Large sample volume is required. 100 to 200 microliter of sample is required. And now we have also produced some advanced or modified tools for detection of different biomolecules into the body that utilize only in 0.1 or 0.2 micromoles of the sample. So this is the disadvantage of ELISA. It requires sample in 100 to 200 microliters. Limited range of wavelengths to read sample. इसमें जो हमारे पास रेंज होती है वेवलेंथ की ELISA के अंदर वो बहुत लिमिटेड होती है. हम इसमें यूवी रेंज में या बहुत ज्यादा हाईली विजिबल रेंज में हम इसको जो है वो डिटेक्ट नहीं कर सकते अपने सैंपल को. So that's all about the ELISA. At the end of this slide you can see a link, or this is the link of that video that I showed you. Okay, I hope you all understand about the ELISA. If you have any query, then please ask me. A vaccinated person will give a positive antibody test, right? अगर वो वैक्सीन जो है वो सही है तो फिर डेफिनेटली एंटीबॉडी टेस्ट हमारा जो है वो पॉजिटिव आना चाहिए। वैसे आप लोगों में से किसी ने वैक्सीनेशन करवाई है कोविड की? कोविड वैक्सीनेशन आपके लिए वर्कफुल है कि नहीं है, इस चीज को भी हम जो है वो चेक कर सकते हैं। अगर हम अपना वैक्सीनेशन के बाद अगर हम अपना एंटीबॉडी टेस्ट करवाएं तो हमें पता लग जाएगा कि वो वैक्सीन हमारी बॉडी में जाके कितना काम कर रही है। सबने ही करवा ली है अप्रोक्सीमेटली, वेरी गुड। जी जनाब, तो एलाइजा से मुतालिक मुझे ऐसा यकीन है कि आप लोगों को समझ में आ गया होगा कि एलाइजा क्या है, इसका प्रिंसिपल क्या है, और अगर कोई कंफ्यूजन है तो आप लोग पूछ सकते हैं। अरे भाई, इतना अच्छा समझ में आ गया कि कुछ भी नहीं पूछना है किसी को। भाई क्वेश्चन ना पूछने की दो वजह होती है, या सब कुछ समझ में आ गया या कुछ भी समझ में नहीं आया। अब मैं क्या समझूं?
हेलो मैम,
जी
सेशन एंड कर दें क्या?
सेशन बस 1 मिनट रुक जाइएगा। एक क्वेश्चन आया है, कैन यू एक्सप्लेन कॉम्पिटिटिव एलाइजा अगेन। देखो बेटा, एक्चुअली सबसे पहली बात तो ये कि आप लोगों को एलाइजा की टाइप्स को इतना डिटेल में देखना नहीं है, आपको सिर्फ इसका प्रिंसिपल देखना है। जो मेजर हमारे पास एलाइजा है वो है हमारे पास डायरेक्ट एलाइजा, ठीक है। अच्छा, अब यहाँ पर बस इनडायरेक्ट, सैंडविच और कॉम्पिटिटिव एलाइजा को हम इतना ज्यादा डिटेल में आपको नहीं करना है, क्योंकि वो फिर आप लोगों के लिए काफी मुश्किल हो जाएगा। लेकिन हमारे पास जो डायरेक्ट एलाइजा है वो हमारे पास ऐसा है कि जो सब सैंपल्स पे, हर तरह के सैंपल्स पे हमारे लिए वैलिड होता है, ठीक है। लेकिन कॉम्पिटिटिव एलाइजा हम सिर्फ उस वक्त करते हैं जब हमारे पास बहुत ही कम अमाउंट में कोई एंटीजन मौजूद है और जो डायरेक्ट एलाइजा से डिटेक्ट नहीं हो पा रही है, ठीक है। उस वक्त हम एंटीजन जो है हमारा जो भी मॉलिक्यूल ब्लड में मौजूद है, वेदर इट इज प्रेजेंट और नॉट, तो हम क्या करते हैं कि हम उस एंटीजन के साथ हम एक सेकेंडरी या एक कॉम्प्लीमेंट्री एंटीजन और ऐड कर देते हैं। कॉम्प्लीमेंट्री एंटीजन ऐड करने के बाद फिर हम उसमें एंटीबॉडी ऐड करते हैं, ताकि उस एंटीबॉडी के साथ अगर एक या दो हमारे पास अमाउंट, एक या दो मॉलिक्यूल भी खाली मौजूद होंगे हमारे पास एंटीजन के, तो वो उस एंटीबॉडी के साथ बाइंड कर सके। तो ये होती है कॉम्पिटेटिव एलाइजा, जिसमें हमारे पास जो हम एंटीजन डिटेक्ट कर रहे हैं उसी का ही कोई एक हमारे पास ऑल्टरनेटिव या कोई कॉम्प्लीमेंट्री एंटीजन हम ऐड कर देते हैं, जो कि समहाउ सिमिलर होता है स्ट्रक्चर में, ताकि वो उस एंटीबॉडी के साथ बाइंड कर सके। वो बाइंड नहीं करता, बाइंड करेगा वही एंटीजन जो कि स्पेसिफिक होगा उस एंटीबॉडी का। Which type of ELISA is most accurate? Definitely direct ELISA. Okay. We will contact you on it.